organism. We have covered already sporulated microorganisms. So we know already Clostridium perfringens is a spore-forming microorganism. It's a gram-positive rod, and it's also encapsulated. It's non-motile, different from Bacillus cereus. If you remember, B. cereus was motile. Now, another difference between Clostridium and Bacillus cereus is that Clostridium is an anaerobe. Now, between Clostridium perfringens and Clostridium botulinum, we will cover Clostridium botulinum later. Clostridium botulinum is an obligate anaerobe. However, Clostridium perfringens may tolerate some moderate exposure to oxygen, but again, it's considered anaerobe. So if you're going to culture or grow Clostridium perfringens in the lab, you need to grow these microorganisms under anaerobic conditions. Sometimes when we have work in our lab with this bacteria, what we do is that we do enrichments and plating, all of that under aerobic conditions, because we know it might tolerate some oxygen, but the incubation always will be anaerobic. Another characteristic is the production of toxins. There are different types of toxins that these bacteria will produce, but it's very specific. These toxins will be produced only during sporulation. So you have to keep this in mind, because this is a very important characteristic of the illness. It's only during sporulation. So the toxins usually will not be performing food. You eat those spores or eat the cells. The cells may sporulate in your GI tract, and during that sporulation process, when the mother cell is slicing and is releasing that spore, is when the toxin also is released. Another characteristic is the rapid growth. It has a very short generation time, about 8 minutes. And I mentioned it's typical of high protein foods. Now let's discuss about the classification of these microorganisms. So here can be a little bit confusing. This microorganism is classified into 5 different types. Okay, so there are 5 different types. Clostridium botulinum, Clostridium perfringens type A, B, C, D, and E. And this classification is based on the ability of these microorganisms to produce different type of toxins. And actually there are 5 type of toxins. There are these 4 toxins: alpha, beta, epsilon, and iota, plus the CPE toxin. I think it's not mentioned here, but this will be our 5th toxin, the CPE toxin. And CPE, this is going to be mentioned in future slides. CPE is the Clostridium perfringens enterotoxin. Perfringens enterotoxin. So 5 type of toxins. The 4 mentioned here are alpha, beta, epsilon, and iota, plus the CPE, which is the Clostridium perfringens enterotoxin. Now, these 5 different types of Clostridium will produce different toxins, but it's very specific from the strains. Typically, the strain associated with the production of CPE is the type A strain that produces this enterotoxin. So every time we mention enterotoxin, it's going to be called the CPE toxin. So one of those the mechanisms of these microorganisms to be very virulent is this prolific production of toxins. So there are so many type of different mode of intoxication from this microorganism, and each type of cell or each individual strain will produce different type of toxins. Okay. In this table, you will find, and it's a good table for you to study, you will find the different type of Clostridium perfringens. So you have here the types or classifications, A, B, C, D, and E, and the different type of toxins, alpha, beta, epsilon, iota, and the enterotoxin. So as you can see, the type A, which is the most common Clostridium perfringens associated with humans, is going to produce alpha and the enterotoxin as well. And here you will have also the genes that are associated with each type of toxin. Now let's talk about the Clostridium perfringens illness. There are two different type of illness. There is this one that is not a very pleasant illness to have, and actually is a very is fatal condition, so people can die from this. It's very rare, not very common. So this one is called the enteritis necroticans or necrotic enteritis, and is caused by Clostridium perfringens type C isolates, and this isolate will produce beta toxin. So this beta toxin will be responsible for this type of illness. Now, the most common illness, this is the most prevalent worldwide, is the food poisoning. So this typical food poisoning will be produced by this toxin that I mentioned, which is the CPE, Clostridium perfringens enterotoxin. It is produced by the type A isolates. Now, this disease, this enteritis necroticans, is not a very common disease here in the US. It's more common in Asia, in Africa, in the South Pacific, and usually affects children with some problems of malnutrition. Is the most common association. And another association which is interesting is related to patients with diabetes. In the US, another interesting fact, in the US so far, they have found, or they have been reported, only 2 cases of these enteritis necroticans. Maybe there might be more cases, but at least if you read the literature, this is what you're going to find. So our most common illness that we're going to be focused during this rest of the lecture is the food poisoning caused by the CPE toxin type A, Clostridium perfringens. There are other type of illnesses produced by Clostridium perfringens. They're not foodborne illnesses, so you can read more about these ones. For example, this gas gangrene, the typical gangrene, is also produced by this microorganism. It's not a foodborne illness, but at least you have the information. So it's just interesting to know how these bacteria can produce a wide array or range of different type of illnesses, and some of them can be critical. Now, this type A, which we associate with the food poisoning, is also responsible for the gas gangrene, but the toxin that it produces, the alpha toxin. So the gas gangrene is associated with the alpha toxin, not with the CPE toxin. There are different characteristics of this microorganism that will make it very that will favor the ability to cause disease. For example, it has a very short onset. Let's mention this onset here for the illness, and also a very short generation time. So the generation time, or the doubling time, is estimated to be less than 10 minutes. If you read the literature, usually books are reporting a generation time of 8 minutes, which is very, very quick. So it multiplies very, very rapidly. And also the ability of this microorganism to grow at higher temperatures is another characteristic that make it more virulent, or to cause illness more rapidly compared to other microorganisms. Okay, we mentioned already the CPE toxin is what causes this typical foodborne illness, and the gene that is encoding for that protein is called the CPE gene. So this is going to be very easy to remember because it's the same, we're using the same letters. Clostridium perfringens enterotoxin gene, or CPE gene, encodes for the production of that toxin. If you notice here, I have placed a little green star here because the information that I'm presenting here in this slide is all very valuable. It's just typical information that you may find in a test. So make sure that you remember everything that we're mentioning here, and also not just remember the information, it's just not memorization, but also make sure that if a case of a foodborne illness is presented in the way that that illness is occurring among the patients, you need to be able to connect that with the microorganism. So it's not only memory when you're studying for these microorganisms. This is very important for you to know. The toxin is made by the microorganism only during the sporulating state. So when the cells are sporulating, and if you remember the different steps or phases of sporulation, there is one of those last steps where the mother cell lyses. It's an autolysis, will release the spore, and during that process it will release also the toxin, and this typically happen inside the GI tract. Now, good news is the toxin is very heat sensitive. It might be destroyed easily at 60 degrees for Celsius for 5 minutes. However, like I mentioned, for us to get sick, we need to consume those spores. And it's not very common to see the spores, the toxin preform in the food. Sometimes that happens, but it's not very common. Okay, let's talk about the characteristic of the disease or the foodborne associated with this microorganism. Let's talk about the onset. Is typically very fast, between 8 to 16 hours, but the symptoms may last only for 12 to 24 hours. So it's a very rapid or fast duration of illness, and it's usually just a severe diarrhea. So it's very well known for causing explosive diarrhea. So what people report is that they go to a restaurant, and if they have eaten a food contaminated with very, very high levels of these microorganisms, and if that toxin was preformed in the food, the onset will be much lower. So people report to have this explosive diarrhea where they cannot wait until they get to the restroom. Typically, there is no fever, very low death rate. Mainly, the most bothersome symptom is that uncomfortable diarrhea. And I mentioned this, we consume the microorganism. The cells will sporulate in our GI tract, and during that sporulation, the toxin will be released. Okay, in here I'm describing some aspects. I have found people being a little bit confused with this microorganism and the fact that it sporulates. So again, keep in mind, you will have a food item, could be meat, even don't just think that meat is the only vehicle. These microorganisms very commonly found in soil. So any food that can be in contact with soil can be also vegetable, or can be any produce or fruits, for example, but most commonly meat. So the food is contaminated with the vegetative cells. Now those vegetative cells, if the conditions are appropriate, they will multiply. So we're going to have a high number of those cells on that food or in the food. Mainly it's just on the surface typically. Now if that microorganism goes under any stressful condition, they will sporulate, correct? And we already learned about sporulation. Let's say that food was contaminated. Instead of vegetative cells, the food was contaminated with the spores. So that meat containing the spores is going to be cooked at a at low temperatures, usually in maybe high or long time. So these cooking conditions will be inappropriate for the microorganism in terms of contamination. For the microorganism will be just perfect. So the spores will survive cooking. If the spores survive cooking, let's say that you're cooking at, I don't know, 80 degrees Celsius, so you have the spores surviving that cooking. Food is then placed in a hot holding oven. This is very typical if you go to a restaurant where they cook prime rib or turkey or roast beef or those big chunks of meat or beef. Usually they cook that early in the morning and they put those meat products in a holding oven. So when they're holding that meat, those spores that survive will become vegetative cells, because we have proteins there and the temperature is fine, and they will germinate, they will outgrow and multiply. Some of them may remain in that food. Hot holding usually occurs at lower temperatures. It's very common to see ovens that are holding food at 40-ish, 45, 47 degrees Celsius. So microorganisms survive that hot holding, and it's going to survive the cooking. They will multiply really fast during the hot holding, and then the person will ingest the food. So you can ingest vegetative cells or you may ingest spores. Either way, those spores that are going to be in your GI tract are going to release the toxin at some point. So that's how the cycle of consumption of food and getting ill occurs and happens. What is the infectious dose associated with these microorganisms? So usually we need to consume very high levels of these microorganisms to get sick. If you are consuming vegetative cells, most likely those cells or many of those cells will be killed during the digestion process due to the low pH. But some of them may be trapped and covered with food and they may survive, and those are going to make us sick. I mentioned a minute ago that Clostridium perfringens is associated with soil. So all of these three spore-forming microorganisms, and actually all of the gram-positive microorganisms that we have studied in this class, they are associated with soil. This is the normal, typical reservoir. So it is common to find soil with levels of 10 to a 3 to 10 to a 4 CFU per gram. Also, it has been reported that meats maybe even contains Clostridium perfringens, approximately 50% of the meat. But however, one good news is that it is estimated that all of that the Clostridium perfringens that is present in food or in soil, just a very low proportion, 5%, will carry CPA gene, which is a good thing because it's highly prevalent in soil. Okay, I'm going to skip this because I have mentioned this already. In terms of epidemiology of this microorganism, how common is this bacteria in food? It has been estimated that is within the top 5 most common foodborne illness in the US. In this report is from 2011, is by this author here, Scallan. He or she mentioned that Clostridium perfringens is top 5 here in this report by the USDA. This is, I believe, from a report created in 2008, I believe, but the statistics are from 2013. Clostridium perfringens was top 6, but still, it's a very highly prevalent microorganism. It's not a reportable illness, and the reason is because the death rate is very low and is a self-limiting diarrhea. Many outbreaks have been reported. This one is one of the most recent that I want to mention. It happened in 2018. It was associated with this restaurant called Chipotle, which I'm pretty sure you're all familiar with this chain. And around 650 people got sick in Ohio in the same chain restaurant. It was due to Clostridium perfringens. So if you have this many people getting sick from the same restaurant, that has only one meaning, which is deficiency in their maybe hygienic practices, and also they use the wrong temperatures for hot holding. So these temperatures that they use here to hold that food are perhaps not hot enough. Hot holding is the most common way for this microorganism to multiply. And keep in mind, these bacteria will multiply at relatively high temperatures. In terms of the control of C. perfringens in food, so usually, or the control and the growth, it will not grow at low water activity. The optimal pH is kind of close to a neutral pH, but the most critical factor here is that growth temperature. Even they have found products where that have been held at 50 degrees Celsius, and Clostridium perfringens still is able to grow there in that food. I want to repeat this again. It has been mentioned during Bacillus cereus presentation and also spore forming presentation, and again I'm going to mention this here. Killing the vegetative cells is very different from destroying the spores. If you have a food that is contaminated with the vegetative cells, if you heat that food at an appropriate temperature, let's say over 60-ish, 65, you will kill these cells. You will destroy them. But if that food contains the spores will not be destroyed. The temperatures and the conditions to destroy these spores are very different, so keep this in mind. I mentioned this earlier, is mainly associated with those temperatures those foods that are temperature abused. I'm missing a D here. And very common to find this microorganism in meat type of product, because these bacteria cannot make 13 of the 20 essential amino acids that it requires for growth. So that's why growing in meat products is perfect for this bacteria. So if you're working with a food processing facility, restaurant, or at home, keep always in mind how dangerous it is to have these food products to be held at hot temperatures that are not high enough. We conducted a study, and I want to mention this just as a proof of what we're mentioning here. We conducted this study several years ago. Actually had the opportunity to work with the USDA during this project, and we visited several restaurants in Nebraska. We visited 3 different restaurants in Nebraska that prepare prime rib. I am pretty sure that most of you or many of you have eaten prime rib. If you haven't eaten prime rib, maybe you have eaten big beef roast or similar products like that. I know that not everyone is a meat eater, but you may be able to relate to what I am talking about. We visited 6 different restaurants. How am I going to forget this? We visited 6 different restaurants, and actually I did this myself. And we collected all of the cooking profile, all of that temperature profile. So when they are making prime rib, they do a searing, which is a kind of just cooking the prime rib outside. And these are the temperatures that we have found here. After searing, they do cooking, and once the prime rib is cooked, they will hold that prime rib. And you can see the extended hours to which this prime rib is being held at the restaurants, between 5 to 11 hours, 2 to 7 hours. So the prime rib is being held at long hours. So what we found is that during that holding time, we found we holding time and holding temperature. So we decided to study two different holding temperatures, 43 degrees Celsius and 49 degrees Celsius. 49 is pretty high. So we inoculated in the lab our prime rib. We inoculated that with Clostridium perfringens, and we analyzed the growth of the microorganisms. So actually we inoculated that with spores. So what happened is that those spores germinated, and so we started with a level of approximately 2 logs, CFU per grams of those spores. And after 10 hours, we can see the levels here. So we have between 7 to 8 logs. So there was an increase of approximately 6 logs. So this is sufficient to make you sick. So, and again, this temperature are the temperatures that the restaurants are currently using. And this is the biggest danger that is associated with this bacteria. In terms of detection of isolation of this microorganism, because it's anaerobe, we have to use always an anaerobic chamber. This anaerobic chamber is a must-have. If you don't have an anaerobic chamber, usually we can use those anaerobic jars that you have seen in our lab. And there are different type of media, and the most common media associated is this TSC agar, and typically we do an agar overlay to help a little with that anaerobiosis. So you do typically pour plate, not spread plate. You pour plate, you have one layer of TSC agar, then and then let it dry, and another layer, a first layer and a second layer of TSC, and this is going to help also with the anaerobiosis, and then you have to incubate anaerobically. And so that's all for Clostridium perfringens.